All right, so this is my project. It is called Microbial Environments and Tattooing. And no, this is not a Star Wars thing. That's just the name of the project that we're looking at today. So first, just to go over a little presentation outline of what we're going to go over. I'm going to be going over the Excalibur and Tatooine project. I'm going to discuss some lipid biosignatures and the astrobiology context of it. Then I'm going to go into some background about lipids and microbes and how they interact with some salts, and then we're going to go over some of what I did this summer. So first, the before I can talk about Tatooine, I have to talk about Excalibur. Excalibur is the overarching mission of why we're including Tatooine as a project. So Excalibur is basically this machine that is being developed to extract molecular biosignatures and lipids, and we're basically going to field test it with Project Tatooine, along with Tatooine providing some under basis for our understanding, but we'll go over that in just a moment. So some of this application is to replicate the sample processing autonomously on Mars. You can see the Excalibur tool on the right here on this picture. Um, this is a draft of the machine, but basically what this machine is meant to do is it is meant to receive the sample, then it's going to grind the sample. It will extract the lipids from it, then it's going to filter the minerals, anything inside of it. It'll concentrate the lipids, and then it'll deliver it for analysis, and then any sort of residue will be discarded and whatnot. So basically, is meant to this machine is basically meant to, sorry about that. This machine is meant to be on Mars essentially with a rover. And our goal is to basically make it autonomously be able to be sent to another planet and be able to grind these samples when a person cannot. So going over Tatooine itself and the YSP program, my main focus was Tatooine, but the overarching project is Excalibur. So Tatooine itself is a project that analyzes microbial communities and factors that affect their survivability and diversity in salty and arid conditions. Arid basically just means the area is dry. These are conditions similar to what we see on Mars, so this is why we have Tatooine as a project itself. So there are these items called IMOMs. They're insoluble macromolecular organic matter. And these are very durable molecular traces of life, and we can use them as biomarkers to establish some working theories that we have that Mars could have been inhabited by some sort of life maybe billions of years ago. And so basically, what we're going to look at is whether or not these certain lipids or organic matter exist on Mars. So these lipids themselves and IMOMs basically take billions and billions of years to break down and go away. So that's what we mean by very durable. As you can see, Kerogen as one of these top sort of substances that we're looking at, it takes about 2.7 billion years to go away. So it's super durable. And we can see Kerogen makes up, on average, a large portion of organic matter itself. So this is why these markers are very important to look at. So for Tatooine itself, we're looking at salt types and aridity, and basically how this influences the biosignature preservation of things such as kerogen in Earth's arid soils. And this is why it makes it a potential analog for Mars, is because of the salt type and aridity of certain areas like this. And then this has applications for the future search of life because let's say certain planets may have had life billions and billions of years ago that we may no longer see anymore because certain things may degrade and whatnot. However, if we could find something like these kerogens or other sorts of IMOMs, we can basically use these as biosignatures because they do not degrade for a long period of time. So we can potentially look at that for evidence of life. So our main hypothesis with lipid deposition is the lipid profile of a microbial community can be significantly altered by the presence of different salts. So what this means is that the presence of different salts within a microbial community can induce hypothetically some sort of lipid change on the profile surrounding it. So the reason we look at lipids themselves is because various forms of genetic material, such as DNA or RNA, are not preserved over a geological time period. And basically, microbes respond to these salty environments and create distinct lipid profiles, which basically means the ratio of saturated and unsaturated lipids shows to be like an example of the microbial stress response to these salts itself. So that's why we kind of look towards these lipids. So some of these salts that we're looking at that we can see similar conditions on Mars are some cosmotropic salts and chaotropic salts. Essentially, cosmotropic salts encourage hydrogen bonding on a molecular level and then increase the saturated lipids within a lipid profile. And this usually stabilizes and promotes the cell membrane. That's why we have the word order up here. It kind of keeps it in order. And an example of this is gypsum up here, which are some of the sites that we will look at. And some chaotropic salts or chaos. These typically break or discourage hydrogen bonding, create more unsaturated lipids, and then destabilize and harm the microbial communities. And this can be seen in usually some halides. So, how do these microbes relate to Tatooine? Well, these lipids produced from these microbial environments surrounding them, lipids can be used as biomarkers when these microbes die, and then analyzing how lipids are changed or preserved with decreasing water activity slash differences caused by these different types of salts. And then, so this all leads to our overall question of what actually happens to these lipids, which is what we're trying to figure out. So, some of the work that I did this summer, I went up to NASA Ames for a little bit and just first with some of our sample work, this is the tool that we will be using to sample some of the places out in the Mojave Desert. I'll get to that in a moment, but these are just some of the tools that we're going to be using. And then I also participated in a literature review that ended up being somewhat inconclusive, but I can talk about that in a little bit. So Project Tatooine, the place that we decided to sample was Bristol Dry Lake in the Mojave Desert, very hot place. This features many soil compositions, including salt compositions similar to those cosmotropic and chaotropic conditions that we talked about. And the Bristol Dry Lake is in a hyperarid and it's very salty. It fits all the conditions that we're looking at for Tatooine, basically. So this is kind of one of our Google Earth maps of some of the sites that we've sampled. So we have the halite areas up here, some mud flats and some sulfate areas, which represent some of those, again, cosmotrophic, chaotropic, salty areas. Um, so yeah, this halite one specifically, as I spoke about earlier, is more representative of being a weakly chaotropic halite salt site. And then this one is a cosmotropic gypsum salt site. So this fits perfectly with this tattooing project of wanting to get these samples of these lipids within these salty conditions because it's an analog for Mars. So some of these sites are being sampled over the course of 4 seasons, which is what the current project is working on. So I helped with the summer portion of this. So this is our sample model. When we went out to these sites, we created this triangular structure, 10 meters apart, and took these 3 different sample sites from each little point. So this gives us an even spatial distribution of the points. And then we also took a soil core depth profile at each one of these blue points right here, which is what you can see me doing right here. This was a relatively easy site. Some of them were not very easy. But yeah, you basically just pump this into the ground and then you're going to pull it out. And then we basically pull the core out and we do some sample work on it. So some of the field test results that we are looking to aim for include pH, ATP, and all these other factors right here. These are just some of the main ones that we're kind of looking at when we're monitoring how these salty and arid conditions affect these microbes and whatnot. This is one of the samples that was probably one of the hardest to get because this is crystallized salt. I believe this was gypsum. And so we really had to dig into the ground to break this layer to get it into our soil core. Did some lab work out there in the Mojave Desert. And basically what we aim to do with this project is we hope to be able to establish a basis of the effect these salt types and aridity have on these microbial communities over time so that we can bring that basis to help us search for life on Mars. So then I did go up to NASA Ames in Silicon Valley a few weeks ago, and we worked on some DNA extraction for this. So some of the chloride and sulfate salts in these soil samples have low biomass and they have some extremophiles in them that are very hardy. And this makes DNA extraction super difficult. So what we basically did up in NASA Ames is we sonicated the samples to wash out the salts, incubated them with lysozymes, and then extracted them. And so some of the things I tested over there or whether these power beads can remain in the sample during sonication, or whether they would cause the cells to lysis or die. This was one of the big things that we were testing. I also ground and sieved some soil samples for some future DNA testing. That will come in handy a lot later. So some of the closing we hope to be able to answer these questions of how salt types and aridity affect the lipid profile of a microbial community. However, we don't have a lot of the results yet. A lot of the literature review I did became inconclusive, or it wasn't measuring the right salt types that we were looking for. So all we can really do is wait on these results to come in and wait to analyze them. So essentially, so after we test detecting these lipids, what's next is we are hoping to have a basic understanding of these stabilization salts on Earth. We're hoping to be able to inform our search strategy for future Mars missions, areas that we can go to that we might want to look for to be able to extract potential DNA or lipids a lot better based on the salty communities, based on how arid the area is. And we also can be able to inform analysis for previous Mars missions. So this is the Curiosity rover that was on Mars, which at some point did include the SAM instrument just to analyze some samples and whatnot, get some data. And we do have that data, but perchance maybe we don't know what to exactly do with it yet until we establish this basis for lipid profiling. And that's very important for us. So not only can we look to the future to see what our future Mars missions should do and look like, but we can also go to the past and take some data from there and try to analyze that with this newfound knowledge that we hope to get. So, anyways, that's my presentation on tattooing. I wanted to give you guys special thank you to my audience here. Special thanks to Anna Simpson for being my PI. And that's about it. Thank you guys.
Awesome. Great job, Nathan. If we have any questions or comments from the audience, you can drop it in the chat or raise your hand. I think I'll start with one about the nature of Mars and the fact that Mars has so many chlorates and perchlorates in the regolith. And given your work on chaotropic salts and these cosmotropic salts, what do you think about preservation of organics in a chlorate or perchlorate rich material like the Martian regolith?
So I believe some of the salt is very important to see because I believe chlorate is cosmotropic, if I'm not mistaken. Um, so basically, it's very important to know this salt type. So I think if it's more building of a community of a microbial community, it can be very helpful to understand that because if it's more building up, there's a higher likely chance of these microbes potentially lasting a lot longer of time. We'll be able to potentially see these lipid profiles as not biodegrading as quickly, and we can see them as lasting a much longer time. So if we can find areas like this, it would be helpful for our understanding and our sampling of these specific areas. I'm sorry, did that answer your question?
It does. So I
Okay.
I think chlorates and perchlorates are on the other side. I,
Okay, yeah.
they're chaotropic,
Mhm.
which is part of why we think maybe some of our results in looking for organics have been skewed because the perchlorates have broken down some of the organic materials. And so, the
Yeah.
research that you're doing is crucial then for understanding not just what might be made by things that could be in the Martian regolith, but how those things have changed over time.
Yeah, exactly. And it can really prove to show how it may not only have these effects of discouraging this hydrogen bonding that's so important to these proteins and microbial communities themselves, but it can show maybe it has even longer lasting effects on examining these lipid profiles over time.
Absolutely. We have a question from Sanjoy in the chat. Curious about how acidic environments as well could affect lipid preservation. Did you read much on pH and the acidity of an environment and how it relates to lipid preservation?
So for pH specifically, we didn't, per part of my literature review, Um, this was some parts that I didn't really get to go to, but the reason we didn't necessarily include a lot about the literature review was because we did find there was kind of somewhat of a positive relationship between pH as it gets more basic between the values of 6 to 9. We found that it did kind of increase these diversity levels, which um, in conclusion, if diversity levels increase, usually you can see your survivability as having increased because you do not need necessarily super strict conditions of survivability. But the reason we didn't really see a relationship were just some of these conclusions from the articles I read were not very significant. And we saw these correlations as existing, but not necessarily super powerful correlations between the two. So that's why we were kind of looking towards measuring this pH with these samples too, and hopefully we get a more stronger correlation with that.
Awesome. Great job, Nathan.
Thank you guys.
I think